Hello everyone. Today we are going to see about production of ethanol. Ethanol is a colorless inflammable liquid. It is spirituous in odor. It is also known as grain alcohol, spirit and gasohol. It is a good solvent for fats, resins, dyes, etc. It can be used as fuel for lamps, stoves. It is also used to run internal combustion engines, and it is also produced from wastes by the action of microbes. Raw materials used for the production of ethanols are sugarcane molasses, cane juice, sugar beets, cassava, grains of maize, corn, and crop wastes. So the microbial action involved in production of ethanol is the involvement of yeast like Zymomonas mobilis, which converts the simple sugars into ethanol. The carbohydrates are found locked in wastes. The complex polysaccharides are converted into simple sugars. Some microbes produce ethanol directly from carbohydrate wastes. Example like Monilius, Fusarium, etc. Microbiochemistry of ethanol fermentation. The carbohydrates locked in wastes are a source of glucose. The glucose molecule is metabolized into two molecules of pyruvic acid, by the glycolytic pathway. In this pathway, the pyruvic acid, two molecules of pyruvic acid, is converted into acetaldehyde. The acetaldehyde is reduced into ethyl alcohol in the presence of alcohol dehydrogenase enzyme and NADH2 as coenzyme. Ethanol fermentation is an anaerobic condition, takes place under anaerobic condition, and the metabolism of ethanol from glucose was first explained by Guy Lussac in 1815 in yeast organism. Industrial production. Ethanol production involves the following steps: formulation of medium, designing of fermentation system, culture of microbes in fermenters, and last step is recovery of ethanol. So first go on to the formulation of media. The sugar concentration of cane molasses and other carbohydrates is diluted to 10 to 18%. The sugar concentration favors the growth of the microorganisms. Ammonium sulfate or ammonium phosphate is added to the diluted medium. So by this, the pH of the medium is adjusted to 4 to 5 by using dilute sulfuric acid, and the starchy media like corn, rye, barley are hydrolyzed by using dilute acid, and they are pumped into the fermenter. So the starch undergoes hydrolysis and yields simple sugars, which are directly converted into ethanol. Industrial production, and the next step is the designing of fermentation system. The fermentation system consists of 3 molasses tank, 3 seed tank, and a fermenter, and last a wash chamber. Among the 3 molasses tank, one is larger in size. It is loaded with large volume of molasses and is named as molasses storage tank. The stored molasses enter another tank where the molasses are diluted properly with water. The diluted molasses enter the sterilization tank for sterilization. So here the medium undergoes sterilization and is to carry out for fermentation process. And the seed tanks are considerably smaller vessels when compared to other vessels. The microbes are cultured in the seed tanks and supplied to the fermenter for the ethanol fermentation process. The fermenter has 3 inlets to receive, and it is has a sterilized medium from sterilization tank, and it receives microbes from seed tanks and other chemicals. It has an outlet to harvest the broth. The fermenter is connected with the wash chamber to harvest the spent broth and transfer to the wash chamber for distillation. Culture of microbes in fermenter. Sterilized medium in the sterilization tank is pumped into the fermenter. Then microbial inoculum is pumped into the fermenter to carry out ethanol fermentation. So the temperature inside the fermenter is maintained by using cold water through the water jacket which is present outside the fermenter, which is coated around the fermenter. Water jacket is present, and the temperature of 330 Kelvin is suitable for fermentation. So the fermentation completes in about 3 days, and the reaction undergoes by the enzyme invertase present in yeast, and which converts the sucrose in the molasses into glucose. Glucose is converted into ethanol by the enzyme zymase. So the chemical formula is C12H22O11, which is the sucrose plus H2O water in the process of invertase enzyme gives 1 glucose and 1 fructose, and the glucose is converted into ethanol. C6H12O6 gives 2 C2H5OH plus 2 CO2. And the recovery of ethanol. Final step is the recovery of ethanol. It is fermented broth in the fermenter is collected from the fermenter and is collected from the wash chamber. It is distilled to recover ethanol. The distillation setup is called coffee still. It consists of 2 fractionating columns provided with perforated plates. One column is called analyzer and the other is called rectifier. Wash flowing through a folded pipe is heated in the rectifier. This pipe releases the contents at the top of the analyzer. Steam is passed to the base of the analyzer to carry the alcohol vapor to the base of the rectifier. The spent liquor is collected from the outlet of the analyzer, and the alcohol vapor in the rectifier is collected through a pipe jacketed with cool water to get 95% ethanol. And you can see the diagram of the analyzer and rectifier. Uses of ethanol. Ethanol is an active solvent of dyes, lubricants, adhesives, some pesticides, paints, explosives and resins. Resins is also used as in some organic compounds and from living things. Ethanol is used in the manufacture of synthetic fibers like rayon, polyester, etc. It is used in the extraction of certain pharmaceutical products. It is used in the manufacture of acetaldehyde. It is used in the manufacture of perfumes, and it is also used in internal combustion engines as a fuel in various countries. So these are about the production of ethanol. Thank you for listening.